Okay, so in the last one we talked about how we are able to genotype polygenic traits and work out how many genes are responsible for a certain phenotype by creating a pure line from a pedigree, crossing that pure line together to produce an F1 generation that's heterozygous for every gene responsible for that phenotype, and then crossing the F1 together to produce an F2 generation, which is going to give us a ratio of homozygote dominant or homozygote recessive, it doesn't matter, but it's going to give us a probability of one of these kinds of homozygotes compared to the general population. And then we're able to use this rule here to determine how many genes are responsible for that phenotype. So like we said on the last one, we cross these together, we've got a ratio here from this F2 of 1 in 64, because the little red box is implying the homozygous dominant, then 62 are different variations of these heterozygotes, and then the little white box is the homozygous recessive. So it's a 1 in 64 ratio. So we have a look here, we see a 1 in 64 ratio, and that equals 3 genes. So we're able to tell that 3 genes are responsible for this phenotype, so this trait. So you might be wondering how we're able to figure out how many genes are responsible. So how we're able to confer this kind of ratio that we get from this F2 cross, sorry, from the F1 cross to produce an F2, the ratio of the F2, basically we're able to determine how many genes are there because it follows a very simple rule. It's basically an nth term, like you probably remember doing in high school. It's literally just 4 to the power of n, so n is just the number of genes responsible. So if we're talking about a phenotype controlled by a single gene, then that would be 4 to the power of 1. It would be a 1 in 4 ratio of homozygotes. If 2 genes, 4 to the power of 2, 1 in 16 ratio. And if you are familiar with doing these kind of Punnett squares for a monohybrid cross shown here at the top right, and a dihybrid cross shown here on the bottom right, the monohybrid cross, this 1 in 4 ratio should be familiar because it's the exact same. So this 1 in 4 fraction should be familiar because you often hear about the 3:1 ratio in a monohybrid cross. This is this would be a P cross, and the organisms shown in the middle are all an fully heterozygous F1 generation. But the point stands, if you were to cross an F1 generation, so you'd have 2 heterozygotes breeding together, you'd get your 3:1 regular monohybrid cross with 3 of the organisms showing the dominant phenotype compared to 1 of those for showing the recessive phenotype. So it's exactly the same as the 3:1 ratio, and this 1 in 16 is exactly the same as the 9:3:1 ratio that we get from doing a dihybrid cross that we would see here. I should have really used different pictures to show F1 crosses, because the F1 crosses get the idea much better. If you want to go into it, just search up F1 crosses for a monohybrid and a dihybrid cross, and it'll make sense because you're getting out of the total 16 possible outcomes, you're going to have one homozygous dominant, one homozygous recessive for the entire 16. It's the exact same thing that we see here. So it's just, the rule is 4 to the power of n, and that's going to give you the ratio that you're looking for that number of genes. We don't really need to go higher than this. As long, if you get a ratio and you like, how do I work out how many genes are responsible for it, just produce something like this and then you'll be able to match it pretty quickly. It's pretty easy to memorize. Well, 1 in 4, 1 in 16, 1 in 64, and then for the others you can just kind of do the math yourself. So for environmental and genetic variants, it should be noted that we have these things called threshold traits that occur at the extreme ends of a normal distribution with high liabilities. So, for example, when the threshold trait is surpassed for the tandem repeat of CAG in the Huntington's gene, when it surpasses 10 to 15 repeats, you get a dysfunctional Huntington's protein that's going to cause the disease. Moving on to the environmental and genetic variants. Environmental factors also contribute to the phenotype of quantitative traits, so when genotype and environment interact. Phenotypic variation can be calculated as the genotype variance plus the environment variance plus the genotype multiplied by environment interaction. We use mean, standard deviation, and variance to understand variation in these quantitative traits. Estimating genetic variation gives us an idea of heritability, which we measure as capital H squared. Environmental variance can be calculated as the standard deviation of the homozygous dominant plus the standard deviation of the homozygous recessive plus the standard deviation of the heterozygous that we get from the F1. So we're getting the homozygous dominant recessive from this P generation up here, and then we're getting this from this F1 generation here. And pause the video if you need to go through a bit more. Genetic variance can be calculated as the standard deviation minus the environmental variance. And then we can calculate broad sense heritability as simply the genetic variance that we've got from standard deviation minus environmental variance, and dividing it by the standard deviation. Moving on to the topic of QT. Actually, no. I'm going to go over this in a separate video, so I'm going to just end this one here because we're running out of time and we have 3 slides left on this topic.